In the name of God, we started together about the study of the embryology. First of all, I want to tell you about the definition of the embryology. Embryology, it's a science that deals with the process and regulations in the prenatal growth and development of an organism or individual in the female genital tract. It begins with the fusion of male and female gametes, that name is fertilization, in the fallopian tube up to the birth as a neonate. Prenatal development involves repeated divisions of the most of the cells in the body, resulting in growth in size, complexity, structural and functional differentiation of the body. Embryology includes the study of startling integration of various complex molecular, cellular, and structural processes that are accountable for the growth and development of a 9-month-old neonate. Embryology divided general embryology. In these courses we study about general embryology. It means it's the study of development during the pre-embryonic and embryonic periods from the first 8 weeks of the fertilization. During this period, the single called zygote is converted by cell multiplication, migration, recognitions into miniature from the one individual with various organ system of the body. In the next semester, we study the systemic embryology, is detailed study of formation of primordia and their structural and early functional organization into various organ and system of the body. Before I started my lectures, I want to review some things about the DNA and the chromosomes. As you know, the chromosomes divided to haploid and diploid chromosome, and each chromosome consists of the molecule of the DNA, and several gene and gene family play important role in the development of the embryo. The number of chromosomes in each cell is fixed for a given species, and human beings it's a 46 chromosome. This is referred to a diploid or double numbers. Okay. During the mitosis divisions, each chromosome is visible, and in the synthesis period, each chromosome in here that show by the simple lines consists of the one chromatid or one molecule of the DNA. You know that the DNA is double strand structures. During the cell cycle, in the S phase, the one chromosome started to duplication. In the 10 of replications is take place, and finally we have a 2 strand of the DNA. Each line resemble of the one molecule of the DNA. Here we have a 2 molecule of DNA, but again we have a 1 chromosome. It means the number of chromosome is 1, and the number of the sister chromatid or molecule of the DNA is 2. After the continuation of the divisions, you can see that more condensation in the chromosome, and each chromatid connected in the midline by one specific structure that name is a centromere. Again we have a 1 chromosome and 2 sister chromatid. In the final stage of the divisions, you can see that we have a split in the centromere, and each chromatid or DNA molecule separated from each other, and finally we have a 2 chromosome and 1 chromatid. The number of chromosomes, as I told you, in human is fixed. We have a 46 chromosome, and we say that the diploid number. However, in spermatozoa and oocytes, the number of the chromosome is only half the diploid number. This is called haploid or half number. It means we have a 23 number of chromosome. After the fertilization, the resulting zygote has 23 chromosomes from the sperms or father and 23 from the oocytes or mother. The diploid number is thus restored. The 46 chromosome in each cell can be divided into 44 autosomes and 2 sex chromosomes. The 6 chromosomes may be of 2 kinds, X or Y. In male, there are 44 autosomes, 1 X chromosome and 1 Y chromosome, while in a woman, there are 44 autosomes and 2 X chromosomes in each cells. When we study 44 autosomes, we find that they really consist of 22 pairs, the 2 chromosomes forming a pair being exactly like. We say that homologous chromosomes. To understand the structure of the gametes and to study how they are formed, it is necessary to first review some facts regarding chromosome and cell divisions. Now I want to talk about the one important process during the embryology. As you know, development begins with fertilization, the process by which the male gamete, the sperm, and the female gamete, the oocyte, unit to give rise to zygotes. In preparation for the fertilization, germ cells undergo gametogenesis, which includes 4 phases. These phases consist of origin and migration, the first phase. The second phase is mitosis divisions. The third phase is meiosis or meiotic divisions. And the last stage, or the 4th stages in the gametogenesis, is a structural and functional changes that name is differentiation or cytodifferentiation to complete these maturations. Now we want to go to the next steps and talk about that first step of the gametogenesis. As you see here, this is one embryo in the 3rd week of gestations. The precursor cells that making the gametes, its name is a PGC, primordial germ cells, as you see here. The primordial germ cells originated from the epiblast layer. The epiblast layer is one layer in the embryo that originated and made in the 2nd week of gestations. The sum of the epiblast cells, especially that cell situated in the tail of the embryo, migrated toward the wall of the yolk sac. This balloon-shaped structure, its name is a yolk sac. During the 3rd week of gestations, the PGC situated in the wall of the yolk sac, you can see in this picture by red shaped structure in here, but this is this isn't the final destination of these cells. It means that these cells have to migrate from here to the posterior abdominal wall. This migration takes place around 2 weeks. It means that around the 5th week of stations, the PGC migrated and entered to the future gonad. It means that next future testis or future oocytes, ovary. That during these migrations we have a divisions. Not only we have a division during the migration, but also during the settle of the cells in the ovary, we have a mitosis divisions. It means that we increase and we have a proliferation in the number of the PGC, because the number of the PGC is not enough, and we have to increase the number of these cells. As you read in the high school, in the mitosis divisions, we have 4 stages. The prophase, metaphase, anaphase, and telophase. In the first steps, it means that in the prophase, the first of all, the chromosome before starting the mitosis is not visible, and gradually the chromosome be condensed and finally visible by light microscopy. And the prophase, usually the longest phase. During this phase, chromosome each consists of the 2 identical chromatid begin to contract and become visible within the nucleus. Centriole move to opposites, duplicated, and move to opposite side or pole of the cells. Short microtubules may be seen radiating from the centrioles. You can see that the microtubules, and nucleoli disappear as the DNA passes to the certain chromosome. And the prometaphase, the gradually nuclear envelope is no longer visible as it breaks up. You can see that here into small vesicle that which this press. Spindle gradually is formed. We say that mitosis apparatus. You can see the microtubule between 2 poles, and the lining of the chromosomes in the metaphase. You can see that the complete formation of mitosis apparatus. This is the centrioles in the opposite poles and the microtubule apparatus that connected the centriole to the centromere. And you can see that the chromosome in equatorial of the cells, and each of the chromosome consists of 2 chromatids. And the anaphase is a very rapid stage, and this phase the centromeres split in 2 and spindle fiber pull daughter centromeres to the opposite poles. You can see that the pulling the chromosome to the opposite poles. Thus, at this stage, the cell can be said to possess 46 pair of chromosomes. In the telophase, the separate chromatid, which have reached the poles of the cell, uncoil and lengthen to form chromatin again, losing the ability to see clearly. Spindle fiber degenerated and the centriole replicates, and finally nuclear envelope reform reforms around the chromosomes at each pole and nucleoli reappear. What's the role of the mitosis? As you know, cell division by mitosis plays following roles. Genetic stability, it means the mitosis produces two nuclei which have the same number of chromosomes as the parent cells. The second role is growth. The number of cells within the organism increase by mitosis, and this is a basis of growth in multicellular organisms. The third role is cell replacement. Replacement of the cells and tissue also involve mitosis. Cells are constantly dying and being replaced, and obvious example being in skin. As I told you, the third step in the gametogenesis is meiosis or mitotic divisions, because we need to the sperms and oocyte with haploid chromosome. Meiosis occurs during the formation of sperm and eggs. It means during the gametogenesis in animals. We have two meiosis. First meiosis and second meiosis. The phase of the meiosis 1 are prophase. That prophase can be divided in following stage: leptoten, zygoten, pachytens, diploten, and diakinase. During the leptoten, chromosomes become visible as long threads attached to each end of the nuclear envelope. During the zygoten, homolog chromosomes, it means that the same chromosomes, the one of them from the parents, the paternal, and one of them from the maternals, pair up. This process is called synapsis or conjunctions. Each pair is called bivalence. This is a bivalent. And finally, two chromatids of each chromosome in the pachytens, forming a fourth double helix called the tetrads. And in this stage started to the crossing over by a one complex protein structure. That name is a synaptonemal complex proteins. The aim of this stage is a exchange of the gene from the parental chromosome. That name is recombinations. In the diplotens, the chromatid separate except in the region of the crossing over, and we have a specific structure. We say that the chiasma formations, and the diakinases recognized chromosome begin to move apart. Each bivalent can now be seen to contain 4 chromatids linked by a common centromer, while non-sister chromatids are linked by chiasma. We have a short metaphase, and the anaphase 1 spindle fiber pull homolog chromosome centromers first toward the opposite pole of the spindle. Unlike the mitosis, there is no splitting of the centromer. The telophase is similar to mitosis. The homolog chromosome which have reached the opposite pole of the cell uncoil and lengthen to form chromatin again, losing ability to seen. the spindle fiber usually disappear, and finally in the cytokinesis, in the end of the meiosis 1, each daughter cells consists of the 23 chromosomes, one number of the copy of the chromosome, each consisting of the 2 chromatids or 2 molecules of the DNA. And the aim of the second meiosis, is important point, is reduce in the number of the DNA molecule. Like a mitosis, we have a splitting in the centromere, and finally we have a 4 haploid cells, and each of them contain a haploid number of the chromosome. It means that 23 single chromosome and 1 molecule of the DNA. As I told you, the number of the chromatid show that the number of the DNA molecules. Okay, this is a third step of the oogenesis, but this is, we have 1 point. The meiosis in the male take place after the puberty, is not take place during the gestational period, but in the female the meiosis is started from the embryonic period but stop and restored after the puberty. This is one major difference between the meiosis in the male and female. Now we want to talk about some abnormality during the gametogenesis. As I told you, sometimes usually the PGC migrated to wall of the yolk sac, and from the yolk sac to the posterior abdominal into the ovary or the testes. Sometimes we have abnormality or abnormal migration during the migration of the PGC, and we have some teratoma. In this picture you see that the oropharyngeal teratoma, this tumor may arise from the PGC or from the epiblast cells, both of which are pluripotent because the PGC and epiblast are pluripotent cells. Tissue within the tumor include derivatives of the all germs layer and may include the gut, bone, skin, teeth, and so forth. And some evidence suggests that PGC, they have strayed from the normal migratory path, could be responsible for some of these tumors. And another abnormality, you can see that the division in the sperm and the oocyte in here, that in the spermatogenesis, finally we have 4 equal functional cells, that name is a spermatid, but in the oogenesis we have only 1 functional, that name is a mature oocyte, and 3 non-functional cells, that name is polar body. Okay, that's again we want to talk about some abnormality here. As I told you, the mitotic divisions, usually we have 4 haploid cells, that each of them contain 23 single chromosomes. But sometimes we have non-disjunctions. It means that abnormality in a separation of the chromosome maybe take place in the first meiosis, like a Bish, or the second meiosis. As a result, the sum of the cells contained more chromosome. It means that we have 24 chromosomes. Some of them has less chromosome. We have 22 chromosome. We say that this abnormality is named as aneuploidy because the normal number of the chromosome is named as euploidy, and abnormal increase or decrease of number, both of them name is aneuploidy. This is a trisomy because if 23 cells, for example, this is an oocyte and 1 sperm with 23 number of chromosome fertilize these cells. We have a trisomy because finally we have a 47 chromosomes, and here we have a monosomy because if we have a 23 chromosomes from the sperms fertilize these eggs, we have a 45 number of the chromosome. The both of them, the trisomy and monosomy, we say that is aneuploidy. Here we have again non-disjunction during the second meiosis divisions. Sometimes we have abnormality during the mitosis. It means that the some of cells doing well mitosis and some of them doing abnormality. As a result, we have a mosaicism. Some of the somatic cells has a normal number of the chromosome and some of them has a abnormal number of the chromosomes. As I told you, we have some abnormality during the divisions, during the gametogenesis. It's estimated that 1/3 of all conceptions in normals, healthy women abort spontaneously. Approximately 1/4 of these occur between pregnancy is detected. Chromosomal anomalies seem to cause about 40% to 50% of spontaneous abortion in those cases in which the conceptus has been recovered and examined. However, many chromosomal anomalies unfortunately allow the fetus to survive to terms, resulting infants display non-random pattern of developmental abnormalities, that is syndrome. One of these syndromes, Down syndrome, is covered in detail in following pictures. Okay, about the Down syndromes, as you know, the Down syndrome is caused by extra copy of chromosome 21 or trisomy 21. The features of child with Down syndrome include the growth retardations, varying degree of intellectual disability, craniofacial abnormalities including upward slanting eyes, epicanthal fold, flat faces and small ears, cardiac defect and hypotonia. These individuals also have increased chance of developing leukemia, infections, thyroid dysfunction, and premature aging. In 95% of cases, the syndrome is caused by trisomy 21 resulting from mitotic non-disjunction, and in 25% of these instances, non-disjunction occur during oocyte formation. The incidence of Down syndrome is approximately 1 in 2,000 conceptuses for women under age 25. This risk increased with maternal age to 1 in 300 at age 35 and 1 in 100 at age 40. In approximately, in here, you can see that in approximately in the 4% of cases of Down syndrome, there is an unbalanced translocation between chromosome 21 and chromosome 13, 14, 15, or 21. Here you can see in this karyotype the correct number of 21, but if you look at the chromosome number 14, you can see that the one copy of the chromosome number 21 joined to the chromosome 14. This is a translocation of the 21 on the surface of the 14. As you see here in the simple picture, this is a 20, the extra copy of the 21 added to above the chromosome number 14. This is a translocation. About 4% of the Down syndrome is caused by the translocation of the 21 chromosome on the surface, another chromosome especially on the surface of the chromosome number 14 or 15. Another abnormality during the formation of the